All right, good afternoon, or whenever you're watching this. I want to do one last wrap-up topic because I want to explain why we have so many connections to the Fred Hutch Cancer Research Center. Uh, of course, it's just across the lake from us. You can walk to it. It takes about an hour. I've done it. But the reason why cancer is important is obvious to most people. The reason why cancer is primarily, or at least fundamentally has a lot of biochemistry research done in it, is connected to this chapter. So I just want to mention this to help you explain why we've already mentioned people at the Fred Hutch. We mentioned that Linda Buck even was involved with her smell research. She came to the Fred Hutch right before she won her Nobel. Why would a smell researcher be involved? Because the same kinds of pathways that signal smell and other senses are also the pathways that sense time when it's time for the cell to grow. Okay? So, and of course, this is a huge issue with cancer. Cancer will happen when the cell's clock gets messed up and the cells keep on growing even when they're not supposed to. This is one of the main ways that you get a tumor. The cell needs a clock because cell division, you have to wait until the right proteins are all in the right place and you have enough of the right kinds of molecules before you move from one stage of cell division to another. When you're making a whole nother copy of a cell, you need to duplicate everything precisely. You need to have two sets of the chromosomes. You need to pull them apart so each cell gets exactly one set. Now, of course, this is a much more cell biology issue, but at its fundamental basis, there is some important biochemistry going on. The different stages need to be timed directly, and cells need to grow. They need to divide. And so there's a lot of kinases that are involved. And in fact, here you have kinases and phosphorylases. The kinases tend to put phosphates on things that turns them on. Phosphorylases tend to take phosphates off of things and to turn them off. And there's a lot of kinases that are just involved with cell division. When something gets mutated there, then you have a cancer cell. When you have a small molecule that binds to one of those, then you have a small molecule cancer therapy. So, and of course, we're going to talk about the underlying biochemistry, which has to do with proteins and phosphates. We're not going to talk about the specific pathways very much. There are checkpoints where the cell sort of says, wait, we need to hold up until we've got everything in order to be able, we've got to have two copies of everything before we can start to divide those two copies up into two cells. It's that kind of thing that's an internal checkpoint. And sometimes it's related to time, sometimes it just takes time, and it's related to number of molecules. The way that this is expressed is through the cell cycle, and you see this all the time whenever you're seeing a cancer researcher talk, because when this is messed up, you get cancer. The actual cell division part is the M phase, where you have mitosis, the nucleus divides, the cell actually divides. That takes about an hour. Then when a cell's in constant division mode, then it will actually keep going after mitosis. It will go through a G1 phase, S phase, and then G2 phase before it gets back to mitosis again. Altogether, these take about 24 hours altogether for a cell to divide. Very rough estimates for just the average type of cell here. Now, normally you want to be able to move in and out of this. When the cell needs to divide, it moves into the cell cycle. When the cell doesn't need to divide, it moves out of the cell cycle entirely to what's called the GO phase, which is withdrawn from this cycle. Of course, when a cancer cell is unable to enter GO phase, for example, it will keep on going through this MG1, SG2 over and over again. The M and the S are where you see a lot of things going on. The S is where you're synthesizing all the DNA, and of course the DNA is essential. You need to have a copy for each cell, and so that's the thing without which you can't have cell division. And so you have resting when you're getting ready to make more DNA, then you have the M where you're splitting apart. And that's really the basic thing that you need to know, is that there are different kind of molecules, and it tends to be the case that kinases are going to turn stages on, and then phosphatases will turn the kinases off, possibly by dephosphorylating them, taking the phosphates off. If you put a phosphate on something, it turns it on. Then if you take that phosphate off, you turn it off. And all you need for a phosphatase is you need a specific binding site, and you need some kind of nucleophile to attack the phosphate to be able to break that covalent bond to the tyrosine or the serine or whatever residue that phosphate is attached to. And so here's an example where you have these cyclin molecules that are turned on and off when you enter different cycles. You have the blue cyclin E, which is with one particular kinase to get you into S phase. And then you have cyclin A, which is associated with the green G2 phase. Cyclin B, with a different kinase, is associated with the M phase. As far as biochemists are concerned, these are all different kinases that turn on different phases. They will each have their own structure and mutations and things like that. You need to get down into what's going on, is getting down to the details beyond what this class can do. But if you think about it, this is where if something breaks, then you can get cancer. There's a complex set of equations. It takes a certain amount of time to make this molecule, to move this molecule over here and stuff like that. And each of these is associated with a particular rate constant, a particular transition state even, and a particular protein-protein interaction. So when something mutates and goes wrong with one of these things, then the cell can keep growing. It can be cancer. One of the ways you can get cancer is if you have a G protein that's supposed to turn itself off, right? Because it has a residue that will hydrolyze the GTP to make a GDP. Well, what if that residue gets mutated? Then you have a constitutively active form of RAS, which means the cell will keep dividing. That would be an oncogene because it's causing oncology, it's causing cancer. But then there's also cells that are like, I'm going to hold back things until everything's ready. And so they stop things from growing. RAS phosphorylates things, keeps things growing, sends a positive signal. There are cell proteins that will actually get in the way of signals. And these are tumor suppressor genes. One of these could be a phosphatase. So if you have a phosphatase that's always taking off a phosphate as soon as it gets put on a certain kinase or whatever, then that phosphatase is a tumor suppressor. When that whole phosphatase is mutated, then you get a tumor because the thing holding back the growth is no longer there. So these are just broad examples of how this stuff can work. You can get pictures like this, and you see how you have the different types of cell cycle, and there are particular kinases and particular phosphatases that are involved with each of these. And actually, so these has oncogenes colored green, tumor suppressors colored pink, and you can see how the oncogenes tend to have Ks associated with them. That means they are involved with kinases, phosphorylation going on. If you have a phosphorylase, it will break the phosphate bond, it will remove the phosphate, and many of these bordered in gray, of course the P here just means protein. It doesn't necessarily mean phosphatase, but I'm sure some of these negative regulators have some phosphatase activity. So let's realize the difference between kinases and phosphatases. But if this means that there's a deep biochemical similarity, and you have some superficial differences in protein-protein interfaces, but you have a deep similarity in where you have kinases versus phosphatases, and you have that kind of thing going on, maybe there are cancers in different types of cells that look very different on the surface, but have similar molecular features underneath. And these molecular features can be targeted by particular single drugs that will work against multiple cancers. So for example, you can have mutation A here is in lung cancer and liver cancer. It looks very different in the different organs, but it's the same mutation. You can have you can have the same colon cancer being caused by several different mutations. And it's the thing of, if you have a similarity of that's at the molecular level, it doesn't really matter if there's a dissimilarity at the cellular level if you can go after the similarity at the molecular level. Looking for these mutations, these particular mutations, and going after them with therapies is what personalized medicine is all about. if it's a mutation in your DNA, you can find it by actually sequencing your DNA. So this actually goes back to the DNA chapter. This ties everything together. And so there's different ways that you actually do these trials, because the traditional way of doing a trial is you assume everyone has the same disease for the same reason. And you put them all together in one trial, and you see how many people. You use different kinds of people. You test it different ways to do phase 1, phase 2, phase 3 trials. But the proposal is maybe we should be, if we're going after a deeper feature, maybe we should be doing our clinical trials differently. Because it's possible that the same therapy could work against different presenting diseases. The idea is known as a basket approach, where you put a lot of people with the same genetic mutation in the same trial. They have different tumor types, and then you separate out how the different tumors respond. Because what you're really testing with the trial is the molecule that you're giving them, is it safe? If you have the underlying molecule you're giving them for different cancers but same underlying mutation, same molecule you're giving them, then it makes sense to do clinical trials differently. Of course, we want to be sure we're doing this right, so this is a lot of medical ethics and a question, but I want you to see where it comes from. It comes from the fact that biochemical similarity can appear in very different cellular contexts. For example, there is a particular larotrectinib, which is a treatment for a particular TRK gene mutation. It's a fusion. It actually happens from chromosome breakages, but it results in different kinds of cancer depending on which different kinds of cell it is. But you could have a therapy that goes after the kinase fusion rather than going after the cancer itself. And so they did a study where they tested the drug with 17 different cancer types. 76% of these 17 different cancer types saw their tumors shrink, and the drug continued to work. It's a tumor agnostic activity. And I think this is cool, and it builds on one of the themes of this chapter. That's why I think it's important. Just remember, when you see K and you involve signaling, it's probably going to mean kinase. And so this belongs in this chapter because it's about a kinase mutation. And they have these different kinds of mutations that they have, which result in different but similar gene mutations, which can result in very different cancers. If you look down here at the very bottom, they have the MAPK ERK, and then they have the PI3K AKT. Those are all things that we've shown somewhere, and we've said those are details. Those will be associated with different kinds of diseases, cell types, and things like that. But I want you to see here they are coming back, and you can really do a lot of work on the cell biology with similar underlying biochemistry. I hope that this works. I don't know how it's going to pan out, but let's keep an eye on this tumor agnostic kind of therapy where you're going after the mutation in the kinase rather than going after the tumor itself. Oh, and there's one more thing I want to mention. For this homework, there's going to be a lot of things that you're going to be looking up in the book. Study the homework. Don't necessarily go back and study the book as much. Study the homework and the lecture as you're getting ready for our final. And you will do well if you can answer questions about that. All right.